All right. So welcome. Up here on stage with me is someone I've had the honor to know for a couple years now, Miss Laura Esserman from UCSF. We're going to build off of what you've heard this morning and take it to those next practical steps that Janet encouraged us to do just now about what does one actually see in platform trials, where they're going, and we'll use the story of iSPY to illustrate that. Later in the afternoon, we'll have many other platform trials, both in oncology and non-oncology settings that we'll talk about in a broader way, but this will be a little bit more of an informal chat and moving forward. So could you welcome Laura for me, please? So, and to warn you, Laura and I were co-authors on a paper called Pipelines about how you turn platforms. So this will be a friendly conversation up here, as we're partners in crime on many of these things. So Laura, not everyone probably knows iSPY in great detail, and they've heard some things about platform trials. Can you tell us a little bit about the adaptive trial you've put together, because it's quite unique, although it shares many characteristics with other platforms?
Well, I think the goal in, when iSPY was born, when I went to Anna Barker when she was deputy director of the NCI and said I was a little frustrated with the way things were moving in the cooperative groups. And I had this idea, and I brought Don Berry with me, and my partner in imaging, Nola Hilton, and Ann listened to us and said, well, we need Janet Woodcock to help us with this. And I think the idea of iSPY was really to try and re-engineer. It's really a re-engineering process. I think everyone who participates in trials from the patient side and the physician sides understands that it's an onerous process. I mean, care is an onerous process, research is an onerous process. They're not integrated, and they're frustrating. And what I think the frustration and the desire to, instead of settling for saying that we could move at this glacial pace, I think this is a clinician's trial. And it's a clinician's trial because all of us who are leaders in iSPY take care of patients. We're on the front line and are tired of seeing things move so slowly, that you have this, great pace of science outside, all these opportunities, and everything coming to this tiny funnel and dribbling in these really arcane ways of testing things that we are just totally frustrated by. So the idea was like, how could you re-engineer trials? How could you re-engineer trials so you actually create a learning system, a learning platform? And you'll hear more about that as how it could come. And there are a lot of things that are required to do that. So the idea is, how could you test multiple agents? How could you surface find and figure out what are the drugs that work best and for whom do they work best? At the beginning, we wanted to adapt within patients, and Don very appropriately said, well, you can't really do that because A, you don't know how to do it, and B, you don't know what the drugs are and you have no idea what they are. And I said, okay, but in 3 years, this is surgeon time, of course, we're going to get there. So 7 years later, we're going to get there. But we are going to get there. That's the point. And so the idea behind iSPY was that we would have a way to, let's face it, let's be honest. Why is there one drug, one trial? Because in academia, that's the way credit is given out. Okay, credit is the currency or the way people get paid. So that's actually pretty stupid, frankly. And so what you need is—
Please feel free to speak frankly to this
right.
group. We're all friends here, so don't hold back.
All right, but really, why would you build this whole infrastructure? It's not like you're not going to, everybody knows what the drill is. We have to see, can we do better than the standard of care? And each disease is going to be a little bit different. There are some diseases where there really are no good options. And breast cancer, there are some reasonable options. They're not great. I like to say, and you give patients their options, no one's like, oh, great, I'd love to do that. But our goal is to have in 5 years, people say, wow, that is so exciting. And we are going to get there. So the idea is to say, well, you could take the standard of care and the standard of care plus, and let's start to see what really moves the needle. And we're not looking for, it's not a trial that answers all questions. It's what is big enough to make a significant impact? What's going to really move the needle? So we can't exclude any benefit, but really, are those the drugs you want out there anyway? No. and we also know, I mean, it was also at the time that everyone say, oh, we want personalized medicine, but then we treat everybody the same. And the other thing is about trying to say, so you're really being thoughtful about what that the disease is different. Then the second thing is, that you've got this 15 to 18 year time, knowledge turn, because, we do everything by phase 1, 2, 3 in the metastatic setting, and then you then operate on people, give the treatments blindly, accrue, wait for everything to mature, and then you say, okay, this is what works. Okay, you cannot shorten that timeline to less than 15 years. So this is like the Andy Grove model of saying, you put out an experiment, you have an early endpoint, and you're always designing the next experiment as you go. And so that was really kind of the idea of iSPY. So the first thing is you have a platform trial. Nobody gets credit for the platform. The platform is there. The drugs come in and out.
And how many drugs are you up to now that are in or have been through?
We're working on our amendments for 14 and 15.
14 and 15 compounds have gone through already, with many more arms than that, I know, because of combos, right?
Right.
Mostly phase 2 focus?
This is a phase 2 trial where we're trying to generate the ideas for how to combine them. But, thinking about, so how do investigators and young investigators, at our meeting at San Antonio, the breast cancer meetings last week, our usual meeting lasting till 12:30 or 1 at night, but people staying to the bitter end, excited about it. Young people showing up because you can be the chaperone for a drug that comes in. They don't have to write the trial. I don't present the data. We let other people, you're finding opportunities for people to be engaged. And so you have to think about how the ecosystem can work for everybody and how the ecosystem works for patients and how you try and say, we had, this is a trial where we've had a lot of input from all sectors. Jane Perlmutter's here today. She's our lead patient advocate, but there are a lot of advocates involved at every step of the way when we make changes and we want to do things. That the idea is that you have this early endpoint. So as a surgeon, I can certainly say that if someone is at risk to die of systemic disease, by going in and operating on them, I am not helping them. Because I'm taking away the biomarker. I have absolutely no idea how it works. And maybe people don't want to see things not working, but that is one thing that makes you want to change. So the critical point was there's been this controversy in the field about whether or not the response to therapy makes a difference. When we started, we got, over the course of the trial, that was one of the things that our industry partner said to us, this can't just be an exercise in finding out which treatments work for which drugs work for which treatments. You have to have an early endpoint, and the FDA has to be willing to recognize the early endpoint. So that was one of the things that we did about 2 or 3 years into the trial. The FDA did come out, we had a meeting and we worked on it, and the FDA did come out with guidance on complete response, but there's been concern in the industry that that's not going to really work for all these new agents. And just last week, we actually presented the data on 10 different agents plus the control with median 2.5-year follow-up, and absolutely fantastic data on PCR. If you get a complete response, if you get complete response, however you get it, whatever subtype you have, you're going to do very well relative to the people who don't. It's an early endpoint, but how did we get there? We were very thoughtful about our denominator. Breast cancer, just like every disease is a range. You can have an allergic reaction that goes from hives to anaphylactic shock. The cancers are no different. They have this range, and especially diseases where you have a lot of screening. You want to make sure that you take out not only your indolent disease, but your disease with late recurrence, because then your early endpoint of complete response doesn't mean anything. But by focusing on high-risk cancer, we really showed that this worked. And the other thing that worked is we have standards.
You've talked a lot about standards.
Standards for how you do imaging. No one gets into the trial until you demonstrate that you can do 5 of the MRI acquisitions in a row, so that change in the volume makes a difference. That we have a way in which all the pathologists are trained and can say, if complete response means complete response, that everyone's, that it's like a choreographed dance. We don't require tons of things, but the things we require are important. And if you think about it, this is the difference between real world evidence and a learning system, right, is that people engage and say, you know what, I want feedback on my response, I'm going to do certain things the same so I can learn. And I would say that we want to evolve to learning systems because you want to continuously improve what you're doing. And that's so iSPY is sort of in between, real world evidence and it's in the evolutionary field of how you get there by people collaborating. The surgeons working with the radiologists to say, where did the tumor start? I'm going to go get that. Working to try and say, what are the standards for axillary surgery or the standards for what is acceptable? And the community really working together because they have the patients in mind and trying to get them to those new endpoints. And we had to introduce a whole new language. What does it mean to graduate from the trial? That means you have, we set the onset it as a standard of 85% chance of a confirmatory trial being successful in a very small confirmatory trial. That means that you're doing your work up front and you are finding that disease group for which that drug works, and so you can do it with a small trial.
And you've now had a number of successes that have graduated.
We've had a number of successes, and a number of them have moved on to phase 3 trials. Some of them, a couple of them have not, partly because of toxicity, partly because the next generation drugs maybe it's a manufacturing problem, the next generation drugs are on their way. But I think one of our goals was to try and do things that the industry could learn from. Everyone can learn. We set it up as a pre-competitive collaboration. So what do we mean by that? That means that industry came to the table, more than one industry partner would put their drugs in, that everyone would kind of learn from what's going on in the field. That's good for everyone, because you don't actually want to spend tons of money on something that's marginal or is going to be outdated by the time it gets there.
Right.
You want to have things that matter. We want to generate options for patients that really matter. And I think it's kind of interesting, our Data Safety Monitoring Board at some of our recent successes said, oh, well, you're finished. You've reached the threshold. I said, really? I said, as far as I'm concerned, we haven't reached the threshold. People are still getting AC and Taxol, and PCRs and 100%, we're not anywhere close to the threshold. There's tons of work to do to optimize combinations and get rid of the drugs that are 30 and 40 years old. I mean, what's the point of the new drugs if you're just piling them onto the old ones that make people sick?
So now you're thinking about regimen change for de-escalation, I think is the word that you use.
Absolutely. So the future of iSPY is de-escalation and targeted escalation. So we're lucky we just got a big program project grant to think about how to transition the trial. I think one of the important things that we did in the trial was we were very thoughtful about how to bring the biomarkers in. Laura Van Veer has been partner in running the iSPY lab and really has a lot of experience in how to get, to follow the steps to get diagnostics used and approved in a clinical setting, which is not trivial, and what you need for that. But we made the critical decision that no one would get in the trial without having a core biopsy and having a successful molecular
profile. So you enforced some standardization on regimen, you enforced standardization on the tests themselves, so every center measures the mutations the same way, as I understand.
Well, but we have multiple platforms, right? So we can look at the immune multiplex, we can look at the expression profiles, we can look at RNA-seq, we can look at phosphoprotein arrays, and it's all there. But we absolutely required everyone to have an expression profile. And it was done in a way that's, CLIA-based. It's, on an FDA-approved platform. And I think the thing that's really important and that's going to come forward is that there's going to be better ways to classify the patients. And we can go back and just kind of rerun all the analyses saying, well, what if we do it a different way? And that will, because we've always done it this way, but we have that data. We now have over 1,200 patients with all kinds of different treatments. And we know the rate of response, we know the absolute response, and we're following everybody. So it's a complete closed loop. We're not sort of testing a little indicator over here and then taking a whole different population and running your outcomes. It's a closed loop. I mean, what other industry works by putting out products and having no feedback on performance and no idea of cost? I mean, nobody does that except in medicine. which is
So
great.
something I'm struck by is it's this closed learning loop, but yet it's also very open because you can start a new arm remarkably quickly, as I recall.
Well, our
right?
goal is if you shake your hand and say you're going to put the drug in and you sign the contract, that we'll have it enrolling in the fastest accruing sites in between 3 and 4 months.
So 3 months from handshake to first person dose.
Contract signing.
Contract signing.
Contract signing. So, but we do a lot
So
of...
lawyers might take 2 years, but once you get past the contract.
No, actually we don't do that either because this idea of the pre-competitive collaboration is that, we don't make special rules for anybody. We don't make them for, there's 16 institutions, there'll be another 5. We've had up to 20 institutions. If you don't perform, you're out. Really, I mean, we give you every chance to succeed, but if you're really not going to succeed, you're out. And everyone's got to pull their weight, everybody. And so when we have a term sheet for institutions. If they say they don't want to sign it, and we say, see you later. Same thing for our pharma partners. We can't change. And the other thing is we don't want that friction to be what drives and takes up time. We don't send the contracts to the lawyers without having an inside champion. If there is no inside champion, it's not going to work. It's a waste of everybody's time. We're not a giant organization. We don't have that. I don't want to, think about it. You don't want your resources to go that way. Who's really interested in going with you to the mat to change practice and take their new drugs and take a risk? And the people who have done that, I think, have been really well rewarded.
Yeah, so you've standardized some science, you've standardized some business process, you've
Yes, a lot
standardized
of business
some
process change, yeah.
legal stuff.
And
If you didn't notice, she also has an
credit.
MBA from Stanford, which is one of my other alma maters, so which is great. So you've really, you started talking about re-engineering, and you really have brought that very disciplined sense to running these platforms, and it seems to be working.
It is working. I mean, people are excited. More and more sites want to join, and I think that as I said, the future is now that we have some success. And I think so. I think at ASCO last year, I was really struck by the idea that we had several trials showing that more is better with a little difference, and then several trials showing that less is better with a little difference. And the truth of the matter is, everybody doesn't need everything. And it would seem to me that the era of running these 5,000, 8,000 person trials has got to go, with these tiny differences, got to go. I mean, what do patients want? I mean, they would love to have a less toxic therapy that's more successful. How exciting is that? And so, people say, oh, well, the patients don't want to do X. I'm like, I don't think they aren't the problem. And the FDA isn't the problem either. It's people's attitudes. It's clinician attitudes and pharma attitudes, and what we've shown is that people are changing, and they're willing to change. They're willing to try new things. So the idea is to start with the least toxic, and if that doesn't work, so it would be worth skipping some of the toxic therapy if you had a chance to have something less toxic that would work. If that doesn't work, you move on to the next phase, and the next phase will be you can get assigned your optimal assigned treatment for your tumor type. And we can compare standard receptors now to these molecular pathways. And then if you get a complete response, you go off to the OR. And last Wednesday, we all voted to drop adriamycin and Cytoxan for people who got a PCR early. That's just such a huge milestone. That's something I promised Jane at the beginning of the trial that we would get to, so I'm very happy about that. But I'm happy about it because my last patient said, please don't make me take the last cycle of AC. It's like death to me. Okay,
And now you have some evidence that for some patients, it's not needed.
Well, we don't exactly have the evidence, but we're going to generate the evidence. I mean, the indication is if you get a PCR, you're going to do great. We don't exactly have the evidence that if you get it without adding the AC, that
it's sustained.
But, it's one of those things you can always give AC later. And if someone doesn't have a response, you can always add it. So I think people sometimes are afraid to change because they're afraid that they're going to miss the opportunity. But I think it's the wanting to give everything is a bad habit, and it's a holdover from all the adjuvant trials where we just gave everybody everything and hoped for the best. And people said, oh, if A is better than B, everybody gets A.
So what I'm struck by is your learning system, and they know that it's systematic, gives them the courage to take away a treatment that if they were more risk averse, they'd say, I'm not sure, but I got to put it in, right? And
Well,
you're saying we
yeah.
can generate the evidence and you can give the community of practitioners there, because you're all practitioners, some confidence
Yes.
that it's not just a one-off risk, it's part of a systematic learning approach, and that you can recover and put in the discipline to be able to do that.
And it's interesting because
that's a phenomenal mindset.
Well, the
Encouraging.
investigators, I think what's happened, what I think is exciting is that we've created a culture of collaboration and we've created a culture of innovation. And, you know, it's not just me. I mean, again, this works because there's so many people that are engaged and working on it. It is truly a team effort. And, you know, it started out as a team effort. It continues as a team effort. You know, I'm a good cheerleader for it, and I'm, you know, help with the system design and move things forward, but it works because everyone's working on it.
And it started with NCI and charitable funding, correct? And you've had some industry sponsorship?
Well,
for
it
this?
actually didn't. It started mostly with charitable funding, actually, and Anne gave me permission to get started when I had what I thought was 20% of what I needed, but turned out to be about 10% of what I needed. You know, I'm an eternal optimist. You know, I'm one of those people that believes that, you know, if you want the parking spot in the front of the building, you have to go at least go there and look for it. So, you know, so I said, meet me halfway. So, you know, Anne said, I said, don't you think we should just get started because success will beget success? And at the beginning, we didn't have a regulatory path forward, and so we just said, give us the drugs, you know, we'll find a way to make it work. And we made it work. And when we got the regulatory guidance, then we started saying we're trying to make it sustainable and have the drugs that come in pay for themselves. But we also have all of this science. We just got this program project from the NCI, which actually will help us pay for some of the science, which is really great. And work with the FDA on continuing to develop a framework for, for the regulatory changes and the regulatory science that's going to have to change as you start to optimize strategies. And you're looking at, the biomarkers are going to be key now. And Anne always likes to say that, we've not made enough progress in biomarkers. But I think these kinds of platforms really help you understand what's going to make the critical difference. And some of it's about science and learning. And then you want to think about what's the easiest thing to take to the clinic. I would say another thing that I'm concerned about, and I think I pressed that in my remarks earlier, is that, okay, let's just say we can get these drugs to the clinic much faster at a fraction of the cost, like 10% of the cost. Is that going to translate into things that are more affordable and better? And I think that we have to think about that.
That's a great segue into the payer side, right? Because you haven't brought the payers in yet on this, right? But possibility?
Well, so I run another big trial called the Wisdom Study, which is a personalized screening versus annual screening. So it's the flip side of the high-risk disease and trying to really understand, screening is a $10 to $20 billion business for one test. So how can we use our resources better, and how can we figure out who's at risk for what disease? We have set that up as a total coverage with evidence progression system, and actually tried, hey, he just brought Blue Cross Blue Shield National Association into this.
So they're helping to pay for some of this evidence development.
The thing about it is it's actually more cost effective to do it in a personalized way, even if you're adding some tests, because you're using that to change your downstream use of services. So in fact, that really is what we are doing with iSPY. And the whole idea is like, and actually, I was talking to Tatiana Powell the other day, and she said, why does all this have to be at the regulatory level? Why can't this be about guidelines and what strategies can work? So what are the strategies that we can think about that would say, you pass certain criteria and then you get into an optimization phase. And the optimization phase, that we get payers to invest in saying, how can I get as good an outcome but reduce toxicity? Because, and who should be engaged in that? And I think everyone has to think about not just what their drug is, but think about the burden on someone. If you can do something in 4 cycles instead of 16, or 4 cycles instead of 8, that's them coming into a center to get treatment. It's the toxicity that's associated, the cost that's associated. And our system I mean, I think this is challenging because the system is built on reimbursement maximization. That's true for the hospitals, it's true for pharma. So like, how do we take the money that's already in the system and kind of repurpose it and say, how can we start to make it make more sense? Thinking about really what's good for patients and how can we start to think about a better system that will reinforce it. So Janet talked about the work that we've been doing with the FDA on these checklists, and you know, my view of thinking about clinical care and research is like, why are there two systems? I mean, what are we practicing for? We should be practicing to get better, but you can't do that if you don't have a system. iSPY gives you an opportunity to keep changing and improving. You know, when you are collecting data, it turns out that everybody needs the same data, and the people who need it most are the clinicians. You need a certain set of data to know you're taking good care of someone. By the way, that's the same data you need for a trial. And the FDA will tell you that 90% of the data they receive for approving an input is not germane to that trial. We need some discipline to say, let's follow these checklists. This is, we've worked with Janet Woodcock and the others, the idea about having a data plan. You don't need everything. And the people who are most afraid of that have been pharma, because it's like, well, what if the FDA asks us X, Y, Z? Well, you start ahead of time and say, do you agree to this? Here's your data plan. Get everyone thinking about it. These are patient-reported outcomes, clinician-reported outcomes. Get these checklists organized so you have your basic hygiene of how you practice working.
So you just suggested collaborative trial design and evidence planning
generation.
And evidence generation with the regulators and with the payers. You just said that.
And with the payers and with your clinicians. This is the way care should be. I mean, let's face it, our care system needs to change. We're using the same tools and standards we used from 1970. You know, that's like trying to take a nuclear bomb and deliver it in a wheelbarrow. You know, in the idea, and I'm sorry, big data aside, screen scraping, we're not going to screen scrape our way into a better way of doing things. I think what we want to do is say...
And for those of you who don't know, many EHRs, the way they connect to the clinical reporting system for the FDA, they literally now have great computer scientists that screen scrape the data, take a photo of basically a screenshot of what's on the EHR screen, and then reinterpret it and do text extraction and re-input it into the clinical reporting system.
You know,
right?
the problem
just mind-boggling.
Well, the problem with the EHRs today is we actually haven't improved the process at all. We're just doing electronically what we used to do on paper. But it's a mindset, skill set, tool set change. It's the idea is that if I'm collecting a certain set of data because I know I want feedback on that, I'm thinking about it. I'm engaging with patients on it. That data, I know that data can be used for a registry. I know that data can be used for a trial. I know that data can be used for some QI project. It's like, we don't have to do it 20 times over. And that's where half the waste is in trials. And the current CRO system, someone coming in and looking for some discrepancy, people hate it because it's burdensome and inefficient. I can certainly see a much more streamlined way of doing things, and we would have a virtuous cycle of improvement and change. And this actually, I'm watching the impact on the investigators and on patients and how exciting it is. And we've evolved to starting to test drugs that aren't combined with chemotherapy. And you see someone coming in, they look like they've hardly had any treatment. Wow, it's such a wonderful thing. People, you know, the patients love it, the clinicians love it. People are coming with renewed energy to want to do things. So we're driving the sense of innovation and demand for change in a much more timely way. I think people have kind of gotten inured to like the bad processes that we've used and just put up with it for a long time. And now that we see, oh, we don't have to do that. We can demand more.
Yes, and so you've painted a picture of great success in individual places for this and this vision, but my sense is it's not all complete yet.
Oh,
right?
no.
There's still a few things left to do.
Just a few.
Right.
So I think there's a couple of ways in which some of this can be, where we can continue to drive innovation. I think one of the exciting things is that as we get better at trying to understand what are the drivers, it's not going to be, personalized medicine isn't giving everyone something completely different. The idea is understanding what are the common things that you have that will drive that will help you make better decisions about what those treatments are, that you learn as you go, and if that doesn't work, you have multiple shots on goal. So we're evolving iSPY again to where it should be. But you want to integrate that with tool systems, these sort of one-source checklists, and we have this sort of collaboratory for accelerating learning in medicine where we can continue to change. But that may be, we know from the TCGA, also the brainchild of Anna Barker, that squamous cell head and neck cancers, for example, have the same biology as basal breast cancer. So why couldn't you add those patients on? Why wouldn't you assume that the same drugs that work for
so we have to scale this up.
It has to be
begin to scale
scaled.
beyond breast, and right now you talk in terms of a dozen or two agents that you're testing, but that's a small fraction of all the agents that are being tested in breast cancer. So we still don't have the scale. This is a unique, done painfully but not yet scaled, and you haven't gone national, right? Your sites are mostly in California and a few other regions?
The sites are across the country.
They're all
They're
across
national.
the country now.
They're national, and we actually have a set of international collaborators that I think for rolling out the confirmatory trials or rolling out, I think it's a little complex for rolling it out, but as we get better and can distill it down, or you want to confirm that your algorithms work.
So it's
especially
a chance
with
to go
payers,
for a global trial. So if I'm a drug developer at a larger firm, I can think of doing soon a global trial on a breast cancer platform.
Yeah, so Don and I developed a phase 3 trial that would adapt to the number of patients where you get early looks. You may spend that alpha, but you're doing it in a way where you're learning. And the thing is, at the breast cancer meetings last week, we saw the presentation of looking at treatment of HER2 2+ disease with the HER2 targeted agents. There were 3,205 patients in the trial, not one whit of difference. Really, did we need to do that? And untold, hundreds of millions of dollars for that. Why are we doing stuff like that today? I mean, it's time to move on.
You didn't need to go through 3,000 patients to figure out that it wasn't really a big difference.
As I said, we could have done it in 50 patients in about 9 months, but or maybe a year. But again, I think people have to say, we can do this. It's a demand issue, that we shouldn't put up with this. Patients shouldn't put up with it. Physicians shouldn't put up with it. No one really wants that. Why is that good? Who wins there? Nobody. And the other thing is, we have a huge problem with cost. Let's start using our resources wisely. Look at that huge pipeline. I look at the numbers of drugs and products that are in the market. One dose, one drug, one thing. We can hardly experiment and do innovative things. Let's make that possible. I think the other thing is, we're actually, a couple of people are coming next year to do sabbaticals, that they're thinking about taking this to ARDS, adult respiratory distress syndrome, and traumatic brain injury, and these are the same ways you think about learning systems. What are the components? A culture of collaboration, finding new ways to give people credit, getting people to understand what's their disease trajectory, where's their early endpoint, what are the early endpoints, what are the late endpoints. How are you going to build systems so that you're getting your data in real time in checklists that feed in, that you don't have to build separate systems and separate silos. This is all a business process change. And so coming up with robust ways of doing that, creating layers like a quality improvement tools that layer on top of the EHR, so that the transactional place and having the data in one place is one thing, but you want to allow collaboration. You want the data. When Ken Butow was the head of this cancer bioinformatics grid, this must have been in 2004. We did this little skit where I was the clinician, and then all of a sudden, like the next day, the FDA had the data at their facility. That was the vision. And we're a lot closer to that now. And I said, okay, 12 years later, we thought it would be a year. Well, that's what I thought I would do. I thought when I left business school, it would take me a year to get all this done. But anyway, as my classmates say, really, you're still working on that?
Yeah, it's still coming.
So annoying. Anyway.
So some questions from the group. It's been an incredible chat and things. Hans Georg, come on up to a microphone, because I think it's easier for everyone to hear you.
Thank you. Great chat, great presentation. The fire is burning down. I heard you say, Laura, that we need discipline with regard to the data we collect.
Yes.
And the majority of data are not useful in the context that the study is about. And I would agree with that. I don't doubt the veracity of that. But don't you think that if we talk about a truly learning healthcare system, we have to think of the future as well? So what is today's hypothesis testing study may be tomorrow's hypothesis generating study. So there were cases in the past where we were happy to have so-called useless data because we could go back to them and see which patients had the adverse event. So where is the right trade-off between what you call the discipline that is economical and it's undisputed, but on the other hand, the investment in the future that what you think is not useful today may be very useful tomorrow?
It's an excellent question. And I would say that there are, I think the framework of our biomarkers is a good framework actually for how to think about things. So in our biomarkers, we have our standard measures. These are for, like in cancer, it's receptors and the 70 gene test. Then we had our qualifying biomarkers. These are things that we think are going to be useful, but we don't know that for sure. Those are your platforms, and they're done in a CLIA lab. They're done with, you're not hypothesis generating, you're hypothesis proving. And then you have exploratory, and not everyone's doing those, and those are questions you can ask. Now you can do the same thing in care, and I would say, people say, well, how are you going to do this? And I can tell you, I'm a clinician. I think our current, I will not name names of what systems, but I would say that our current systems are how I know I'm going to be successful. People hate using the electronic record systems today. I mean, I spend 2 hours documenting at the end of the day, and what do I have for it? Nothing. I don't get feedback on performance. I can't transfer anything. That rage and that, that is fuel for me. I know I can beat that. So, so that makes the bar very low. So I think you can say, well, what are the key things that I need to do to take care of someone, And it's not staging systems and all those are derivative. Get the basic data you need. Plus, everyone can say, oh, here are the things that we want to collect. And individuals can then add things on, and you don't need that on everybody. But, that can be generating data in the background. We could do so much better than we are. And I'm not saying that that's a good way to start, and then you can keep adding on. I mean, we did this in iSPY. We did it in such a way that I think they're going to, as I said, I'm sure we can do a better job of classifying tumors and optimizing that. But we had to generate this data and have it all together to be able to do it.
So one of the great benefits I've seen in talking with Laura on this is, as you bring this new mindset into the clinical trial data is really out of the EHR and the form of the practice, you're bringing much more quality right into the EHR so that every patient is becoming more and more an audible learning data point is what Hans.
And I have to say, you inspired me.
We need a couple more quick questions.
Yeah, I have to say, you inspired me by telling me that if PCR really turns out to be such a good endpoint, put it out there and just wait and see if people do well 3 years later, and that's good enough. So I'm thinking, oh, that's like so awesome, right? Because then that really, I think the FDA, the EMA, the regulators would be much more willing to put drugs out onto the market or products onto the market if they knew they'd get feedback on performance. And then you don't have to make it so onerous and so risky. So it's, again, to make the world better, everyone has to change.
So that's great. We're going to do 4 quick questions here in the next 7 minutes and break for lunch at 5 before the hour. Question on the left.
Yep, I'm Bob Campbell from Brown University, and you mentioned some of the enablers for the iSPY platform, the prognostic enrichment, the ability to define an early endpoint and have it validated with regard to the relevance of long-term outcome, and also the, let's say, ethics of a neoadjuvant approach in terms of treating patients prior to removal of the tumor. Are there lessons from the research investments that went into that for how to approach that for other diseases?
Yeah, I think I can talk more to you at the break, but I think, at the beginning, trying, the mentality someone comes in is, oh my God, a tumor, I just have to cut it out. And, and if you're the only one saying, oh, you should start with chemo or neoadjuvant therapy first, and no one else is saying that, that's a big distance. But I think any good example of saying, wow, look how they've done this and made progress, that you know, that you just say, you know what, the order of therapy isn't what matters. It's understanding what works and giving yourself a chance to be armed with information to change. It's the conversation that you have with the patients. It's making clinicians feel comfortable and having that and realizing, wow, I can change, but then you have to be willing to do it. You know, you can do much less surgery. You can improve people's
Said
lives.
by a surgeon here,
Absolutely.
right?
And the thing is, and you use your craft wisely, not just using it. And you give people their life back. If you say, oh, you know, your tumor's gone, you have like a 5% chance of recurring. When they come in the door, it's like 60%. And you know, that being said too, when we look at people who have significant residual burden after triple negative breast cancer, and they have a 20% chance of being alive at 2 years, if that isn't compelling reason to do something different, I don't know what is. So that's the motivation. You know, don't wait. Let's do it now.
Excellent. So you've had time to, again, John, shorten up that question. Thank you.
The iSPY experience, it's tremendous. We would learn so much. Can you tell me about adaptive randomization and what we may have learned from that? Because always the criticism is simple randomization, you control for known and unknown factors. Did we learn anything about other factors?
It works. Okay, it works. You know, I don't know, the basic thing is like, why it works and why it's efficient is because the next person that comes in, you know, why should I give, I mean, one of our drugs didn't work in HER2 negative patients. You know, after about 6 or 7 patients, we stopped giving it to them. You know, it's like, that's the point of Bayesian statistics. You can improve your prior probabilities and move on. And so you don't get trials of simple randomization, 3,205 patients, no benefit. I submit to you, not a very good plan. And it works. And the thing is, and the biomarkers don't work at the beginning, but because you've got unequal randomization, but now we have 1,200 patients, now it does. You got to stay the course and let the whole system learn. Now we're going to do something different where we're going to start working on optimization. If this doesn't work, you move to the next. If that doesn't work, you move to the next.
Excellent.
Stay tuned.
Dan, and then back to the right.
Dan Hanley, Baltimore. Can you tell us when and how you're integrating patient-reported outcomes into this platform?
So we now use quality of life. We use patient-reported outcomes as quality of life. We are in the process of developing tools to help us with toxicities. Turns out the way we capture toxicity is pretty terrible and not very informative and not very patient-centered. We just got a grant from the Bose Foundation to really integrate this. We have a year or two to really do that. And you know, we have the tools. We collaborate with Salesforce, so that's been a tremendous partner for us to help learn how to develop some of these tools. But we also, I mean, we're using rapid prototyping and Google Forms and trying to test what's working. And you know, this is something that I wish we could do faster, but the great thing about this is going to really help us communicate better with patients. And you know, we haven't done as good a job as we wanted in getting all that information back to patients, even though we intend to, because things get ahead of you. But Jane's going to keep us honest, and we're absolutely going to do that. I mean, everybody who's participated in a trial deserves to have feedback on how their arm worked. Certain information should come back. I mean, the more data you can give back to people, the more people own it and want to participate. This business of why, if your patients are really partners, treat them as partners. And you learn from them, they learn from you. And you know, the other cool statistic is if we looked at all the patients compared to controls, we've doubled the chance of people getting a PCR in the trial compared to controls. Not that we can guarantee anyone any outcome, but when you get people engaged and picking the right things and moving forward, being in a trial, you're getting potentially tomorrow's better therapies. Today, everyone should want to do that. Who wouldn't want to start with the least toxic and have multiple shots on goal? Why wouldn't you? What you want to do is design a trial that everyone would say, wow, I want to be in that. And you yourself would want to be in that, right?
Well, final question.
Hi, I'm Heather Canavan. I'm from the University of New Mexico, and I'm actually a breast cancer survivor. And I really was, thank you, I didn't really have anything to do with it. It was people like you who really helped a lot. But this really appealed to me because as someone who works in this area, I would have thought that I would have had a little bit more of an understanding, but it's not. Even if you have a PhD in this area, it's still terrifying and kind of overwhelming. And I like this idea of having the patients be really involved with getting the correct kind of care and the thought you've put into this project, and I really applaud you for that. The question that I have is, I like that you're focused on let's go for the less aggressive, less toxic versions, but will you allow also for the people who are like me, who are just thinking let's go for the thing that I know is going to work, and having a little bit of patient selection, because speaking as someone who is in that seat, kind of the only part of the process you have any control over is the final decision of this is the one I'm going to go for. And I think if I had had that removed, it would have been really overwhelming. And so I wonder if you've got that built in of a little bit of patient choice as well, or if it's taking that out of that.
We'll talk about it. I think the idea is, if you knew, would you be willing to try something new and less toxic,
Right.
where you're monitored every 3 weeks and then 6 weeks, and then if it's not working, you move on to what you know is the best treatment, not the old treatment. I mean, if you want to do the old treatment, you can get that outside of the trial.
Right.
But to give you what is the best treatment according to your subtype and give you the maximum chance of a good outcome. And then even there, if you don't get there, we'll give you yet a third chance and we'd randomize to something else. Nothing stopping you from, I mean, what I say is, if you don't want to do that, we can give you standard therapy, no problem. And I would say the last innovation that we're looking at is, based on the data we presented at ASCO with the immuno-oncology drugs, and the response that we got, we tripled the chance of a response. I have to say, I as a clinician, I have trouble. I myself am not at equipoise, and what I'm working with the FDA is say, we've got this historic database. Do we need to keep giving people control? We have lots of controls now. Do we really need to start doing that? And Rick Pastor said there's the idea of, contemporary, that you can have concurrent controls or you can have contemporary controls. Don's worked on some innovations called the iSPY 2 time machine where you can compare. But I think the point is, and I think this is, Ansgar, this is one of the points you made, if you get somebody to your endpoint and you're driving everyone to PCR, our goal with iSPY 2 Plus is to say 90% of patients to a complete response without standard chemotherapy. That's a great goal. And if you get them there and you do the follow-up and less than 5% of people recur, isn't that the goal? How do we make those frameworks possible? I
And
can't
learn and get product approvals and get the payers to be
I
helpful
refuse to
on
believe
regimen optimization.
that doing the right things for patients, that there isn't a science behind it, we can't get the regulatory science to follow, and we can't make it right for the system. Because more is not better. More is just more. This is a very American notion, right? And in the name of wanting to do the right thing, we have overtreated a ton of people. We are not helping the people at risk by overtreating them. If someone doesn't get it and wants to do that, they can do whatever they want. They always have that choice. But in the trial, our goal is to try and figure out how to get people to the most effective treatment with the least toxicity. And if we don't try, we're never going to get it. If we just give them the kitchen sink, that's what they're going to get. And I also see people at the end of the day suffering from lots of complications that it just makes me want to tear my hair out, that I wish I could go back and fix.
Great.
Thank you very much.
And can we thank Laura for a great discussion? Thank you for the chat. We're a few minutes late, but lunch will be in the room next door, and Michael Cusimano will be talking in about half an hour or so to us about what we can learn from other industries, not just healthcare, about adaptive platforms. Thanks so much.